Now we're going to move on to Deb's presentation, which shows in vitro and the method development.
Okay, so I'm coming from India, from a institute which is here, right beside the seashore. So today I'm going to talk about a newly discovered pathway of neurotransmission, which is an additional pathway that we recently discovered. So to start with, how the signaling in our brain occurs. So the signal from one neuron goes to another neuron through a region called synapse. What happens in the synapse is that there are some molecules called neurotransmitter molecules which are packed in these vesicles, and they get released from one cell, and the other cell's receptors accepts those neurotransmitters. Okay? And this is how the signal has traveled from this neuron to this neuron. Now, in this whole process, the only action of the neurotransmitter is to bind with the receptor only. So this is the so-called receptor-mediated signaling pathway of neurotransmitters. Okay. However, people have often forgotten one of the major component in this whole diagram, which is the lipid membrane. The synaptic vesicles as well as the plasma membrane is made up of these lipid molecules which are amphiphilic in nature. Now, if you look at the structure of some of the neurotransmitters, those are also amphiphilic in nature, and that's why we found that these molecules can also bind to the lipid membrane, and not only binds to the membrane but also changes the property of the lipid membrane. So next we asked whether this binding interaction and this changing in property of the lipid membrane also happens in real neurons. So the question is, if you have in a neuron there are many vesicles having different amount of neurotransmitters in it, and the concentration is very high, like it's 300 to 400 millimolars. So does these neurotransmitters bind to the vesicular lipid membrane and change its property? And now, now this lipid membrane property is a very important thing in biology. For example, if you look at the exocytosis process itself, so this vesicle comes and fuses with the plasma membrane. So it's a membrane-mediated process. And now, if this neurotransmitter binds with the membrane, it can, and then changes the membrane property, then this process, you would predict that this process can also get affected. So this is the question I am asking, whether the concentration of serotonin in this case changes this exocytosis process. And first of all, we like to question whether it actually changes the membrane property of the vesicles in live neurons. So how do we do that? Okay. So the idea is, you look at individual vesicles. It's a live neuron. You look at individual vesicle, and then you label the lipid membrane with a dye called Nile red, and the spectra of this dye changes with membrane polarity. So this dye measures the polarity of the lipid membrane, and this is quantified by a ratio called green divided by red ratio. Okay. Now, what you do, you also measure the serotonin concentration of the same vesicle. But the challenge is, this you can do, Nile red you can do by normal confocal fluorescence imaging, but serotonin you cannot image so easily. And on top of that, you need to do both of these things simultaneously, because you need information from the same vesicle. So that led me to develop a microscope which can detect serotonin and Nile red simultaneously. And you can see these images, where this is Nile red image of the cells and the corresponding serotonin image. Okay. Now, if you look at two vesicles, the red vesicle having higher serotonin, you see the Nile red spectra is red shifted. Okay. Now, if you compute the green by red ratio and measure it across many cells and many vesicles, and you see that the vesicle having higher serotonin has a low G/IR ratio, which means the vesicle having higher serotonin is getting more polar in nature. Okay. So what is happening? If serotonin is going inside into the lipid membrane, water is also going in and it's making the membrane more polar in nature. Now, if you look at if you actually see the exocytosis process, it involves first association of the vesicle and then it fuses. So now in this state, if there are two membranes like this and water goes in, it's an unstable state because the water is like captured. So what will happen, these two membranes will come and associate with each other so as to leave these water molecules free. Okay? So can we measure this thing in, in actual vesicles? So how do you measure that? We did total internal reflection fluorescence microscopy. So what you can measure here is you can measure individual vesicle. You can see the vesicle come in, bind with the lipid bilayer, go out, or it can also fuse, that you can also see. And then by imaging this process, you can measure each of these rates. And what we find is that if you increase the concentration of serotonin, the rate of unbinding decreases. Means the vesicle is staying much longer on the bilayer if there is more serotonin. And now that leads to a increase in the total rate of fusion. Okay? So then we asked whether this thing is leading to any consequences in the cell. So what we did, we measured the vesicles G/R ratio of these neurons. So you get a G/R distribution of the vesicles. And then you trigger exocytosis of these vesicles. Some of the vesicles will get released, some will stay. And then you measure the G/R ratios of the vesicle which are staying, which has not undergone exocytosis. And what you see here is that only this part has undergone exocytosis and the vesicle having high G/R has not undergone exocytosis. So in conclusion, what we say is that in a neuron, if there are many vesicles and the vesicle having higher serotonin has a more polar lipid membrane, and therefore these vesicles are actually preferentially exocytosing. Okay? Now, not only exocytosis, we have also seen that if you have the serotonin molecules in the lipid membrane, it can affect other processes. For example, activity of proteins or membrane proteins, and these proteins are not related to serotonin. These are like very unrelated, which has no interaction with serotonin. So what happens? We look at this receptor. It's a GPCR receptor, Y4 receptor. When serotonin binds into the lipid, so we see the activity which is measured by the ligand binding strength of the protein, and that ligand binding strength decreases if you have more serotonin in the membrane. So it's a membrane-mediated allostatic effect of serotonin. Okay? So now, the conclusion of this talk is the take-home message is that I am saying that serotonin can also signal through the membrane, not only through the receptor, and that it can do by two ways. First, it can interact with the vesicular membrane and control or tells the cell that now this vesicle is filled and now you can undergo exocytosis. Second way is even after undergoing exocytosis, it can bind to the lipid membrane, change the activity of many membrane proteins which are there near the exocytosis region. So this is the second way how serotonin can signal, and both of these are through the lipid membrane. Okay? Now, if you now we have seen that biologically molecules are there which are affecting the cellular functions through lipid membranes. Now you can design molecules chemically, like which is not present in biology, so it will be bioorthogonal, and then you can use it as a drug and like affect the cellular functions through the membrane. So this is a new strategy of drug discovery that I am proposing here. So with this, I would like to acknowledge all my lab members, especially my supervisor, Professor Sudipta Maiti, and all the collaborators who are involved in this work, and thank you.